Good evening. This is your trusted news reporter from Channel A&W News, Andrew Alvey. Today, our attention turns toward the novel coronavirus and how it's affecting our nation. As of this minute, 257,000 people have died from the virus and over 3.5 million more are sick. Tonight, we'll be talking to some of America's most renowned scientific experts as they educate us on the specifics of the virus and let us know what is being done to combat the pandemic. First, we turn to Professor of Biochemistry at Cornell University, located in the state most impacted by the virus, New York, to tell us a little more about how the coronavirus replicates and forms inside the cells of its host. Dr. Gibson?
So using the mechanism for coronavirus, we'll be talking about a mechanism in action for the remedies. So first, we need to talk about the S spikes on the coronavirus. The S spikes are a protein encoded, one of the 4 that are encoded by the coronavirus genome. These spikes are very important because they will not only help bind to the receptors, but they can also cleave and allow for the genome to enter into the cell. So this new coronavirus takes one of 2 different forms. The first one is the primary one, which is the one that was found in the MERS virus in 2012 as well. So basically what it's going to do is when trypsin and TMPRSS2 are present, those are 2 different types of receptors. When those are present with the ACE2 receptors, the viral membrane and the host membrane will fuse together and allow that genome into the cell. The second pathway, which is the endocytosis pathway, those S proteins are going to bind to the ACE2 receptors and then through endocytosis enter the cell as a whole. Then, because of the low pH in the cell, cathepsin L is going to become activated. This is going to break down and cleave that S2 site protein, allowing for that genome to escape into the cell. Now, once the genome is into the cell, all the steps are similar for both fusion pathways. First, that genome is going to go to the endoplasmic reticulum, where those S proteins are then going to be biosynthesized. In the endoplasmic reticulum Golgi intermediate, it is going to take that genome and those spike proteins and basically create a new viral cell. This viral cell is then going to be transported out of the cell using exocytosis.
Thank you, Dr. Gibson. That was very informative. We appreciate you coming on tonight to inform the public. Next, we'll be turning to another expert in the field, Dr. Madison Lett, a research scientist from the World Health Organization, to help us understand more about how the virus replicates and forms inside host cells. Take it away, Dr. Lett.
While we don't know much about coronavirus, we are learning more every day. For example, we're learning how the virus replicates and forms in the cells of its host. The coronavirus contains a genome composed of a long RNA strand that acts like an mRNA when it infects a cell. The genome directs the synthesis of 2 long polyproteins that have the machinery needed by the virus to replicate. When replication occurs, it makes more RNA, several structural proteins like virions, and 2 proteases. The proteases play an essential role by cutting the polyproteins into all these functional pieces. Most of the cuts are made by the main protease of coronavirus. The main protease of SARS-CoV-2 is a dimer of 2 identical subunits that form 2 active sites. Its folding shape is very similar to serine proteases, but a cysteine amino acid and a nearby histidine perform the protein cutting reaction. It also has an extra domain that helps to stabilize the dimer and a peptide-like inhibitor bound in the active site. SARS has 2 proteases. It has the main protease and the papain-like protease. The main protease is very similar to that of the coronavirus, but the peptide-like protein is different. It has a single subunit and uses a cysteine in the reaction. 3 specific cuts are made in the SARS polyproteins by the papain-like protease. It also clips several proteins in the infective cell, specifically ubiquitin from ubiquitinated proteins. This removal causes an interference with the production of interferons in the innate immune system. Interference causes our body's defenses against viruses to fail and allows the coronavirus to infect and replicate within our bodies.
Thank you, Dr. Lett. We are very grateful for the work you are doing at the World Health Organization and for taking time out of your busy schedule to talk to us tonight. Lastly, we'll be hearing from Dr. Willow Dove, a physician at Grace Sloan Memorial Hospital in Seattle, Washington, who works in research for finding treatments for diseases such as the one we are currently experiencing. Dr. Dove, can you tell us about possible therapeutic techniques that you and your colleagues are currently working on?
Of course, Andrew, and thank you for having me. Our most promising possible therapeutic technique so far is that of human antibodies taken from the blood serum of those who have survived and recovered from the virus. We had heard rumor that work was being done in China, where the plasma of recovered people was being used as a treatment, and decided to look more into this here in Seattle. We began working on a more specialized form of treatment, where we extracted specific antibodies from the plasma of recovered patients to test it as a type of therapeutic technique. These antibodies work to neutralize the effectiveness of the COVID-19 virus by binding to the spike protein that enables the virus to enter human cells. This research is still in its preliminary stages, however. We must be able to identify which antibodies are most effective at treating this virus and whether there should be different antibodies used depending on whether the treatment is being used as a preventative measure or to help those who have already been infected and have presented with symptoms. Once these antibodies have fully been determined, however, it is possible for us to engineer these so-called monoclonal antibodies to be made in larger quantities. On the plus side, the antibodies even may have some immune boosting properties. However, there is still much we don't know about the protective immunity of COVID-19 since it is so new. Practically speaking, there are issues such as the fact that monoclonal antibodies have been historically difficult to make and to administer on a large scale, since they have to be injected or administered via intravenous drip. They also tend to be high cost and thus have low availability in non-wealthy countries. If this therapeutic technique pans out, there will be definitely more roadblocks in terms of mass manufacturing, distributing, and cost. We will let you know as we learn more. Thank you, and stay safe.
Thank you, Dr. Dove, for keeping us up to date on the current research into therapeutic techniques. We have for you a short clip now showing exactly what the human antibody bound to the coronavirus spike receptor looks like, as Dr. Dove described. I'd like to finish off today's broadcast concerning the coronavirus by quickly reiterating how important it is to wash your hands. Although it seems like a trivial and unimportant step, it is actually very effective in preventing illness. The coronavirus is encased in what is called a lipid envelope. Detergents such as soap can break apart this fatty coating, rendering the virus ineffective. Not only does it disarm the virus, scrubbing your hands is a mechanical action that allows you to physically rub the germs off your hands and rinse them down the drain. A simple action such as taking care to frequently and efficiently wash your hands can help to protect not only your life, but the lives of everyone around you. That's all we have for tonight on this evening's broadcast of A&W News. Thanks to everyone for turning in, and be sure to wash your hands, stay educated, and stay indoors. This is Andrew Alvey signing off.